Well, first of all, I'd like to thank you and thank the organisers for inviting us to speak at this this meeting. It was really been an amazing meeting, seen some fantastic new technology, and as Moe said recently, I think how do you say, transition to industry. So this is one of the final academic talks that I guess I'll give, at least for the moment, and it's a real honour to follow Albert and talk to you about something that I've been working on for the past 3 or 4 years. Whilst in Carol Robinson's group at the University of Oxford, and that's trying to see whether we can push beyond measuring mass using native mass spectrometry, even at the sort of unprecedented level that Albert just described, but also get some structures from native mass spectrometry as well. And when I was preparing this talk, I kind of was thinking about, I spend a lot of my time now looking at small molecules bound to structures, but how we use structures to inform our understanding of biological processes in the world. And it dawned on me that we're actually looking at protein structures every day at the moment. So this is just some images that I picked from newspapers, broadsheets from the UK and from your side of the pond. And when we look at these images, they share a common feature, which is kind of, I guess, ubiquitous at the minute. And what we're really looking at here, of course, in all of these images, is a structure of something that we can't see with the naked eye, and that's the coronavirus. And the most distinctive feature, I guess, are these spike proteins that we've all been talking about. And I kind of wanted to highlight how interesting it was that we use structures not only to understand the molecular detail of things that are happening, but also communicate how proteins are important to biological function. And so how does native mass spectrometry fit into all of this? Well, one of the most obvious, and I think, honestly, lovely examples of this to date is some work from Albert. I'm not just saying that because I spoke after him. I'm speaking after him, where, I mean, structure, we've shown over the past few days, or you've described over the past few days, how cryo-EM and high resolution structures can really give us fine detail. But there are limitations to the technique, and one of the limitations is dealing with heterogeneity. And that's really what I want to talk about as we go through today. And this is a really great example of how native mass spectrometry can help with heterogeneity. So here, what Albert's group were doing here was looking at a dynamic assembly in the circadian rhythm, and they're interested in a complex that appeared over time and disappeared over time. So by using native mass spectrometry and profiling the time points, they could find the best conditions at which then to perform the cryo-EM and get their structure. And I was sort of inspired by this, I guess. I wanted to give you, in the first half of this talk, a little vignette of something that I've worked on whilst in Carol's group. So this is using native mass spectrometry to determine the structure of a really interesting bacterial nanomachine called the injectosome. And I was really privileged to work on this with a very talented PhD student, now a postdoc at Imperial in London, Lucas, and Professor Susan Lee, who's moving to the US to set up a cryo-EM group at NIH Frederick. I encourage you to check out their website or check her out. She's a great collaborator. So what were we doing with this? What is this export gate in the injectosome, and what were we trying to do? So I have a bit of a soft spot for bacterial nanomachines from my PhD work, and they are quite amazing. So it turns out that some pathogenic bacteria have these extracellular protrusions that you can see on the right of the slide here, these needles, and these are called these are part of the type 3 secretion system, or the injectosome, and they use these needles to puncture the cell membrane of their host cells, which could be us, and inject things, and those things can be proteins and other biomolecules. And here in the center you see an electron tomograph and a structure, a combined structural map of this large nanomachine. And you can see how it spans 3 membranes, the inner membrane, the outer membrane, and then your membrane. And right in the core of this machine, on the inner membrane of the bacterium, there is this structure here. And you can see in the kind of cartoon representation that many of the structures of this machine have been solved. And I think we saw some of those on the first day of this series. But this part highlighted in red, called the injectosome core complex, or the export apparatus, is really key. It sits on the interface of the inner membrane and the cytoplasm. So everything that goes through this needle has to go through this assembly. And we were really interested in finding out the structure of this assembly to see if we can then start to impact the function of this, and perhaps provide an avenue for for antibacterial agents, et cetera, et cetera. So it turns out that this complex is quite enigmatic. It's a complex of 3 proteins. I'm going to call them P, Q, and R. The nomenclature, as always in biology, is extremely complicated, and depending on the bacterial system, they're called other things, as you'll see in a minute. I'm just going to call them P, Q, and R. And they're all together in an operon. And in the literature, when we started this project, there were some conflicting reports, not on structure, but also on stoichiometry. So we had some results from native MS suggesting that this assembly was a complex of 5 P subunits and 1 R. So where Q is, no one really knows. There was some quantitative proteomics suggesting that it was a 5 to 1 to 1 complex, but also evidence to suggest that perhaps that wasn't quite correct. And then we have this cryo-EM structure of a 6P complex. So I guess what we really wanted to do is we wanted to get our own structure and see if we could kind of resolve this mystery. And there's this great paper by Laurie Pasmore describing the cryo-EM workflow. I won't go into it in any detail because we've had many discussions over this workshop about this. But essentially what I'd like to highlight here in red are the difficult bits. So these are the bits that cause you problems if you're a structural biologist. And all of these red squares essentially can be perturbed or can manifest as problems if your sample is heterogeneous. And the worst point is if you've done your gel, so that's in the first part, and you see the bands of the proteins you expect. You've got a nice peak on your sec. Your negative stain looks okay, but of course you don't have the resolution to really find out what's really in there. Your cryo-EM screening might look okay and things look okay. And you go all the way to step 4 where you're acquiring thousands of movies, spending a lot of time and effort. And then you find that you've got one of your subunits missing and not enough images to get a structure. So thankfully, there's a solution to that. And unsurprisingly, you might say, we believe that was native mass spectrometry. And what we can do is we can slot native mass spectrometry in here into this workflow between steps 1 and 2, and we can do some screening with native MS. And as Albert said, it's worth pointing out here that, native mass spec has undergone a bit of a revolution over the past 5 years. It's not only for specialist labs now, it has become more robust and has become routine. And I'd like you to think about that as I go through the next step. So this is a membrane protein complex, and as I said, 3 components, P, Q, and R. And it's worth saying at this point that we're pulling down this complex with a strep tag on the R, and that's important as we go through. So, your standard biochemical workflow, we're expressing this as an operon. We lyse, extract the cells, pull the complex down. We perform a sec step. And then I want to highlight this step here that may not be obvious to everybody. When you're performing membrane protein native mass spectrometry, you might want to buffer exchange into your non-volatile salt so you can perform your native MS, but you also have a choice here of the detergent that you use for the native MS step. So initially what we used is this detergent, which is a sort of happy detergent, DDM, that the complex was purified in. And when we did the native mass spectrometry, we got a spectrum that looked like this. So it's not really important to go through and identify all of these peaks, although we did, or rather the PhD student Lucas did. But essentially what you need to know here is that firstly, to get this spectrum, we had to use extremely harsh MS conditions under which the assembly was dissociating into bits. And even without that, we knew that there was some heterogeneous things going on here. So what we needed to do is see if we could improve the native mass spectrometry conditions to get a cleaner spectra. And anybody that's been through the literature knows that perhaps DDM is a detergent that requires quite a lot of energy to remove inside the native mass spectrometer. So we went to one of our go-to detergents, and that's C84. And when we did that, we got this spectrum here. And straight off, you might say, well, that's great. It looks a lot less heterogeneous. You've got this nice distribution in the center here. You can see this sort of pink hexagon. And this is that fly P hexamer that I mentioned the structure of earlier on. So on the first glance, this looks very nice. However, remember, we're pulling this complex down on a strep tag on the R subunit. So something's gone wrong here. And this is one of the things that isn't really touched too much in the native mass spec literature, but exchanging the detergents for native mass spectrometry at this step can also introduce rearrangement. So clearly we get a nice spectrum, but something that's not very physiological. It was nice because it allowed us to perhaps explain why somebody else had seen this hexameric structure of the hexameric P. But really, we wanted to get something with all of the subunits to determine the stoichiometry. So we did a full detergent screen. And we went through, and I kind of call this like the Goldilocks scenario, where we have to try and find the detergent that's just right. And as we went through this class of detergents, these polyethylene glycols, we found one right at the bottom, C85, where the spectrum was much less heterogeneous. And I'll show you that now. And here, yes, you can see some adduction onto the peaks, but we don't have multiple stoichiometries. And here we see a stoichiometry of 5 to 1 to 1. So this was very encouraging. But when we analyzed this spectrum in detail, we also saw some little peaks that corresponded to 2 Q subunits, which suggested that maybe this wasn't the end of the story. What we did then is we went back and we had another look at the purification. So we've gone through this cycle once. We've gone through the expression, the purification, the initial native mass spec. We've optimized the native MS buffer. What we then did is went back again to the expression and tried to extract the protein in different detergents. And when we did that, you get the result that you see here. So the only thing that's different here is that instead of extracting the protein in DDM, we're extracting it now and purifying it in an LMNG. And I don't know whether you can quite see it, but down in the bottom here of your gel, you can see for the band corresponding to Q, what we see now is a much darker band here. When you do the native mass spectrometry, and I've deconvoluted these spectra, you see a difference. In the DDM sample, you can see 3 major species with 2 of the smaller Q subunits. With the LMNG sample, there are more Q subunits that appear. What we did is we prepared grids at the same time of these samples and performed some initial cryo-EM screening. And what you see is this. So in the DDM sample, you see a top view that looks a bit like a circle, and a side view that looks a bit like a rectangle. And when you do the same thing in LMNG, we get a much higher resolved picture, and I'll show you that in a little bit more on the top view, and a slightly longer side view. And what this is telling us that in these conditions, we think that the native mass spec has shown us the most intact conditions, or the conditions where the complex is intact. What we did then is we pushed that through a standard cryo-EM workflow, took those grids, acquired movies, and we got this lovely structure on the right-hand side. So this has been published in NSMB, and the structure is like a camera lens, and it always amazes me to see these structures, particularly of small complexes. And we've got it here in the closed formation, in the closed state. Obviously, in the bacterium, this would be open to allow proteins to move through. And taking structures from multiple different organisms in the same way, that allowed Susan's group to come up with an assembly mechanism for this export apparatus. And you can see here how the nomenclature is different. And this is how we think that the 5P, so this is where the nomenclature is different. The 5P subunits are forming a scaffold, recruiting the R, and then the S here corresponds to the Q. And then that sits at the heart of this export apparatus. Future work on this project is trying to get this assembly into an open state so we can look at the rearrangements between closed and open. And I wanted to highlight this because whilst it's not using completely new technology, it's ready for use. And really, there's no reason why not to use native mass spec at this point in the cryo-EM workflow to minimize your time, minimize your cost, and hopefully maximize your success in obtaining these structures. And it also works for membrane proteins, as I've shown here. In the second part of the talk, I really want to focus on work that I've been doing for the past 3-4 years now with Stefan Rausenbach, who you can see in the bottom left. Firstly, when he was at the MPI in Germany, and in Stuttgart, and latterly at Oxford. So he's now there at Oxford. And if you've seen him talk before, you'll have seen some of his previous work using soft landing to look at small molecules. And I'd like to highlight also the contribution of these 2 guys on the left-hand side, Paul, who's a DPhil student, and Tim, who's a Marie Curie fellow, a postdoc in the group. Paul and Tim are, you'll see their full names at the end, they are exceptional students who will both be looking for jobs. Well, hopefully not Tim, but Paul soon as he completes his PhD. Keep an eye out for them. And I'll show you a little bit about what we've been doing. And what we've been doing here is combining native mass spectrometry and this thing called soft landing in order to prepare samples for single particle imaging. And also I'd like to highlight the contribution of Sabine, who's also transitioned to industry. We did a lot of this work with at the beginning, and the guys from Thermo Fisher, particularly Alexander, who's believed in this project right from the beginning, back when not so many people did and thought it was a little bit crazy. So why would we want to do this, and what is native mass spec and combined with soft landing? So we've shown over the past few days how powerful cryo-EM can be for structure, but there are still limitations, and a lot of these limitations tend to focus on heterogeneous samples. And I just wanted to highlight 4 examples of heterogeneous samples here. First, when we get unknown density. So this is a membrane protein, and in the ligand binding pocket of the structure of this membrane protein, they saw some density, but they couldn't resolve it. So they didn't know what was there. Another example, it was clearly there, but the reason why they probably couldn't resolve it is that this was a low abundance species, and this is a particle averaging process. So as the thousands of images were combined, or the thousands of movies were combined, any assemblies that contain this density tend to average out. Now, there are algorithms to cater for that, but often, and as I said, this was published relatively recently, can't fully resolve what's in there. This middle case here is a case of 2 proteins that are stuck together with some lipids. And this is a crystal structure. And in the center of this crystal structure is this red patch. And this red patch corresponds to a lot of heterogeneous lipids, but none of them are resolved. In fact, only one of the lipids is resolved. We've got the case of small proteins. So I know there are amazing strides in cryo-EM for tackling small proteins. I think I may be wrong, but I think the limit at the minute is streptavidin, or perhaps even smaller, around 50 kilodalton. But the guys that are performing this routinely tell me anything less than about 100 or 150 kilodalton is challenging. And structures from complex mixtures, and I really want to highlight this. I mean, most of the samples that we've seen and the structures that we've seen have come from highly homogeneous samples. And wouldn't it be great to be able to get structures from highly complicated samples? And I guess the nearest we've managed to get to that is pushing all the way to the cellular context and performing tomography. And each of these cases, this heterogeneity, and it was great following Albert, because I guess he highlighted this to the extreme. Mass spectrometry is fantastic for picking out heterogeneity. And what we really want to do, and this is the challenge that we set ourselves, is can we routinely use native mass spectrometry to prepare protein assemblies from mixtures and image them at the true single particle level with sufficient resolution to derive useful biological information? So that's a very long sentence, but essentially you can see a native mass spectrum down at the bottom there of a part of an E. coli lysate. And really what we want to know is, can we get a structure of that? So this peak here, whatever the ion here, or the molecular entity that corresponds to this, can we get a structure of it? And can we get the structure of it from this? So can we pull it out of this assembly and get a structure? And that's really what we've been trying to do, and it's quite challenging, as you can expect. But what I wanted to do to show you today is not go over the work that's been done previously, and there is a lot of work that's been done previously, also by some of the guys at PNNL, but give you our take on this process and some areas that we found interesting or challenging in order to do that. But this is still our aim. I'm not going to get to the end of the talk and show you that we can do this, unsurprisingly, but I want to tell you about some of the things that we've found on the way. So combining native mass spectrometry and soft landing. I don't need to go through the native mass spectrometry process, but essentially, for those that are slightly unfamiliar, we can take heterogeneous mixtures of proteins, ligands, and assemblies or complexes, ionize them, exchange them into a non-volatile buffer, ionize them by native nanoelectrospray, transfer them into the gas phase of the mass spectrometer, where we can mass select out individual assemblies. So we can throw away what we don't want, and we can measure what we do want. And if we can do that, we can mass filter this ion beam. And at the minute, we can detect them in these immensely sophisticated and sensitive detectors, such as the Orbitrap. But what we could also do, maybe, is we could deposit them onto solid supports. And if we can do that, we have a way of teasing out or pulling out of complex assemblies the thing that we're very interested in, which could be a drug bound to a protein assembly, or it could be a small protein assembly in a complex mixture. And the way that we've decided to tackle this is by modifying that amazing mass spectrometer that Albert just described, the UHMR. And here you can see on the left-hand side a picture of the instrument as it was pre-COVID. We've moved this to another room to increase our COVID capacity. And highlighted on the right-hand side, you can see that the base instrument, so that's in blue, but with the cover off. And on the back here, we've added this deposition stage. And the deposition stage is currently a work in progress, but and I want to talk to you a little bit about what that is and what that allows us to do. So going to a more schematic kind of visual, this is the instrument in the form that many of you will know. And what we've done is at the end of the HCD cell, we've removed the Faraday cup that's here to measure charge, and we've added a vacuum chamber with a gate valve. And I want to talk to you about two of the key parts here. So firstly is this lens array here of electrostatic lenses, and that allows us to take the beam as it comes through the instrument, through the HCD cell, and port it out through the gate valve. It allows us to focus the beam and directionally change the beam. So we can change the beam, we can move the beam in all directions. And then right at the back here, we have our sample holder, and this is a lovely, elegant design of sample holder from Stefan. And an important feature of this sample holder is that it allows us to do lots of things. So we have an energy detector, which I'll come back to in a minute, and we can also pop different substrates in there for imaging. And essentially, if it fits in the hole, we can land on it. So here are two examples. So this is a solid support. So this is a HOPG carbon amorphous carb, sorry, HOPG graphite support, so atomically well-defined support for AFM. And we can also do this with the EM grids, and we can do them with the EM grids mounted in various different ways. So when we did this on this instrument, you might say, well, I mean, did it work? Could you get any ions from the front to the back? And in fact, that was our initial challenge. And what I want to do is talk to you about some of the observations we made in terms of transferring the ions from the beginning to the end of the mass spectrometer. And what you can imagine is if you want to do that, what you might want to do is measure the current at different stages in the mass spectrometer. And the commercial instrument isn't set up to do that. So what we did is went through extensive modifications, and this was mainly the work of mainly the work of Paul and Tim, to add current measurement devices to each of the key stages in the mass spectrometer. And that allowed us to follow the ion flux through the instrument. And if we were losing ions, find out where that was. And this is the sort of profile you get. So this is a profile of native BSA. So native BSA, and I've normalized it to the initial ion current. And what you can see here is if you start off with 100%, at some point, it really does drop off a cliff. And at the end, what we could do with in the original version of this instrument is we could get about a current on a good day with the wind behind us. We could get a current onto the sample support here of about 13 picoamps. What does that mean? Well, without going into the calculations of how many molecules that is, that sort of gives us an hour deposition based on experience. Now, if you want to change parameters and you want to image rapidly, an hour deposition isn't that great. You really want to be changing that by an order of magnitude. And this turned out to be an extremely challenging thing to do, and could only really be done through the hard work of Paul and Tim. And we went through an extensive characterization of the entire instrument from back to front. And unsurprisingly, at the end, what we found to some of you that build instruments is that the most gains were to be made here. So if we could increase the brightness of the ion beam in this region by an order of magnitude, the rest of the instrument was so sophisticated and so sophisticated we could continue that through the instrument onto our sample. So we made some modifications to the source region, notably increasing the size of the orifice and changing the pumping. And when we do that, we get a curve that looks like this. And this may look relatively simple and relatively straightforward, but really I can't highlight enough what a major achievement this was, because it's a game changer in terms of optimizing parameters. We haven't even started to image yet. We're just looking at ion signal at the back end. So now for native BSA, at any rate, we can move from an hour deposition to sort of a 10-minute deposition. And of course, that depends on the coverage, and I'll come to that in a little bit. The next thing I want to talk about is the deposition itself. So as I mentioned, we have this amazing sample holder at the back. And so the beam passes through, and this is a schematic of the ion beam you can see in the top right here. And we have this energy detector. And what the energy detector allows us to do, so using this electrostatic lens, we can bend the beam, or we can point the beam either at the sample or at the energy detector. And when we point it at the energy detector, we can measure a current at the back here. And what we want to do is we want to measure the energy of the beam. And why do we want to do that? Because we want to soft land, or we want to deposit without smashing the molecule apart. Some of you in the audience are very familiar with SID, or surface-induced dissociation. We do not want to do that. We don't want to crash land. We want to soft land. So what does this look like? So this is actually a signal for denatured BSA, so that's why the current is very high, so over 300 picoamps of the energy detector. So what we do is we ramp the voltage, or we change the bias on a grid, a retarding grid here in the center of this energy detector, and we keep increasing the voltage until the signal measured at the back of the, on a plate at the back of the energy detector drops. You can see that here, and flatlines. What we can do here is take the first derivative, so that's the midpoint of this inflection, and we can measure the width. And this is the energy of the beam with a width profile here. And when we land, we want to land at an energy just sufficient for the proteins to slow down enough to gently land, but not high enough for them to smash. And why is this important to have such a refined measurement? Because surprisingly enough, different proteins behave very differently. So these are two energy profiles for denatured BSA on the left and GROEL on the right. The first thing you might notice is that they require different biases for the soft landing, or different biases to be applied in order to land softly. And secondly, the width of the beam is large in the case of denatured BSA and not in the case of GROEL. And if you're interested about why that might be, we've worked on that a little bit too. I'm happy to answer some questions about that. So I've described how we can get higher signal currents, which are necessary in order to do the experiment, and then measure the energy of the beam for different proteins. So BSA is relatively small. GROEL is about an order of magnitude larger. I'll come back to those proteins in a little bit. But what about imaging? So we have two imaging modes. The first is low resolution screening, and the second is high resolution structure determination. And you know, some of you might be getting impatient and saying, well, where are the structures? Show me the structures. At the beginning, when we were starting to do this, we couldn't really afford time to go onto cryo-EM microscopes, especially if we were just going to image pancakes. What we wanted to do is see whether what we were actually depositing with our soft landing skills were globular assemblies or not. And we decided that we would do this using AFM, because AFM can be extremely useful. And when you image what you've deposited, so this here is a deposition of a protein, a globular protein, onto a solid graphite sample, you see something like this. So on the left-hand side is what we want. So this is each of these individual blobs is an individual protein assembly. And you can see at the top here, the height profile of those assemblies here. On the right is what happens when you really focus the beam tightly, or you land for a very long time. So this allows us to investigate the deposition time and the beam focus and the spatial properties of the beam. And this is extremely important when we want to move to single particle imaging, because if you have something that looks like this in red, you're never going to be able to pick out your particles. Whereas if you have something in blue, you will. And something to note here is that you can see that the height of these particles is relatively reproducible. And I want to just go into a little bit of detail about that. So it turns out we can do this with lots of different assemblies. So at the top here is some data from GROEL and a height distribution of deposited particles. So GROEL is an 800 kilodalton assembly. At the bottom here is a similar thing for a membrane protein, and a membrane protein outside of the micelle and a membrane protein inside of the micelle. And what we can see here from the height distributions is that we see differences. So GROEL is larger, as you might expect. And OMPF without the micelle, here in yellow, is smaller than OMPF with the micelle, as you can see in orange. And that might not be very surprising, but that was an amazing result for us, because it told us that we don't really know very much about the structure of these assemblies at this point, but we do know that they're globular, so they're not unfolded, and the trend seems to correlate with size. And when you do this with a number of assemblies, as I've done here, and plot them on a graph, so this is a graph of height against a molecular weight that's to the power third, so it's linearized the graph. What you see is you see a sort of vaguely linear trend of height with size. And if you overlay that against a spherical model, so that's just assuming a constant density for the protein, you see that we undershoot all the time. And we were interested in understanding that. And if you look in the top right of the slide, just something to note. So AFM height measurements are extremely accurate in the Z direction for height, but extremely inaccurate in the lateral dimension. That's because of convolution with the tip. That might be obvious to some, but we're very confident about these height measurements, not so confident about the sort of XY measurements. And I won't go into this in detail because I guess there's an extra 10 minutes talking about it, but we investigated this in quite a lot of detail, and Stefan performed a lot of calculations, and the group in MPI Stuttgart actually modeled this. So what we find is when you deposit onto solid substrates, so thick graphite substrates, you get a van der Waals compression of the molecule with the solid support due to the interaction. I just wanted to mention that at this stage, and note that as we measure onto amorphous carbon or onto TEM grids, our support is a lot thinner, and so we don't expect to see this compaction as much. Happy to talk a little bit more about that, but that's really Stefan's work. So moving on to high resolution structure determination. So okay, we can deposit stuff. It looks the assemblies look intact. They don't look like they've fallen apart, and their size or their height correlates with what we might expect from an intact assembly. But can we get some more information? So really what we want to do now is we want to ask, is the architecture of these assemblies conserved? So, you know, if this is a protein assembly, is it all jumbled up, or are all the proteins where we left them when they left the solution? So to test that, we use this very large test molecule cryo-EM. So it's a 14-mer. It's a barrel-shaped protein. Some of you from the native mass spec community, it's a bit of a blast from the past because people have been using this molecule for a long time. When you measure it by mass spectrometry, it looks like this, so very clean. And when you deposit it using native mass spectrometry and then image it using AFM, you get something that looks like this. So in the middle is the control. That's a blank TEM grid. So we can perform AFM on the TEM grid. Very difficult, but you can do it. On the right, you can see the TEM grid after soft landing. So we see lots of particles. And what we wanted to do is look at the architecture. So we performed what one might do in a lower end electron microscopy experiment. So we performed negative staining. So we landed, performed negative staining. So this is the grid on the left after soft landing in the instrument. So taken out of the instrument and imaged. And then on the right, after being taken out of the image, out of the instrument, stained and imaged. So you see a bit of aggregation from the stain, but you see some particles there. Note, you can't really compare the densities because there are different points on the grid. So what does this look like? So this is cryo-EM. On the left, that's a drop cast sample. So just a sample prepared from solution, has never seen the mass spectrometer. On the right is what we got when we went through the mass spectrometer. And if you zoom in onto some of the particles, you can see this. So what this told us is that this was actually our first attempt. So this is we call this K4. So this was the grid that it came from. It was our first attempt, I think 3 years ago now. But what this showed us is, yes, there are some particles that have broken apart. Yes, there are some damaged particles, but some of them have survived. And some of them have retained the architecture, and it's extremely clear to see it. You get side views and top views of the cryo-EM, much like you would from the control. So now moving on to results from Oxford. So we then said, okay, you know, we want to move on quickly. We don't spend a long time looking at cryo-EM. So, you know, you're going to laugh now when I go to the next test molecule, but we wanted to ask, we know we can retain architecture, but what about morphology and structure, internal, external structure? So we moved to ferritin. So ferritin, again, is a very large assembly, a bit smaller than cryo-EM, so more like 400 to 600 kilodalton. And when you spray ferritin by native mass spectrometry, you get a spectrum that looks like this. And you get two distributions. This distribution on the left with resolved charge states corresponds to apoferritin. And on the right, this blob corresponds to hollow ferritin. So ferritin is an interesting molecule because it's a sort of sphere, and inside the sphere, it can either have an iron core or not an iron core. And it's also a favorite of the cryo-EM community. I just want to highlight this on the right. So these two papers down here, these are the, you know, some of the papers from this year showing sub 1.5 angstrom resolution. So where you can see the hydrogens of cryo-EM, they do that on ferritin. So we want to, you know, I'm not going to show you that type of resolution yet, but I'm going to show you what we can get when we deposit ferritin using our Orbitrap mass spectrometer in Oxford. And when we do that, what we want to do then is do some class averaging. So I'm not going to go through this slide too much, but essentially this illustrates what we're doing. This is a reference sample. This is not deposited sample. So we will go through, or Tim will go through manually. He will pick particles and he will perform 2D classification on those particles using RelyOn. And this again is what you should expect. And I'd like to highlight this here. So this is negative stained. So you can see a dark halo around the outside. That's the heavy metal binding to the outside. And this dark blob in the middle is the iron core. This is where we're at the minute. So on the left is a drop cast reference. On the right is the same solution that's passed through the mass spectrometer. And again, we can see spherical objects, not quite at the resolution you can from Dropcast, but with one of the really lovely things that we can see here is that firstly, they're the right size, so they're the right dimension. And secondly, we can clearly see the iron core. So we can see exterior surface features, what we want to do next, and we can see a little hint of internal features. We were really buoyed by this, so we want to push on again to the next step, and that's where I'm going to stop. And I'll show you just one image from the next step. So the next step would be to perform some cryo-EM. So this would be to deposit ferritin and perform cryo-EM to see if we can get some internal features. And only at this point can we start to evaluate what our soft landing procedure has done to the internal structure of the molecule. And I'm going to leave you with this as an image. So this is an image of ferritin that's gone through the mass spectrometer. It's been M/Z selected. It's been deposited on a grid, plunge frozen and imaged. And this is a single particle. So those of you that are used to looking at grids, I don't know, I was very excited by this. So this is Timessa's data. What we're doing now is extending this, acquiring the images that we need to start to build a structure, and we're progressing in the right direction. COVID has slowed us down significantly, but I think that this is an exciting result considering it's been through the mass spectrometer. So in conclusion, what I wanted to say is that native mass spec, it can add enormous value to current cryo-EM workflows. It can take a lot of the pain away from preparing grids and finding out what you've got at the end isn't really what you thought you got on there. So use native mass spec. It's ready to be used. And it's also especially useful for looking at fragile or dynamic assemblies that perhaps are difficult to understand how they behave. I hope that I've sort of shown the first tentative results that preparative native mass spec or soft landing offers potential to enrich and prepare transient low abundance assemblies for imaging from complex mixtures in the future. We've modified and optimized this Orbitrap UHMR profile for beam brightness. We have full beam control for soft landing and short deposition times. Negative stain TM suggests we can retain the overall fold, and cryo-EM allows integration of internal structure, and watch this space in that regard. Another important thing which I didn't mention here that we've been working on with the guys from the Klaus Kern's group in Max Planck in Stuttgart is that this deposition is compatible with low energy electron holography. And for those of you that don't know this technique, I would definitely suggest you go and look it up. Low energy electron holography is extremely exciting. We've got some very nice results there. It's a true single particle technique with no low molecular weight limit, and theoretically less than 2 angstrom resolution from a single particle. And we have some interesting results there that we'd like to show later on in the year. So in summary, I'd like to thank you for listening, but also Carol and her group for a fantastic 6 years there. Stefan, with whom I still collaborate as a visiting scientist because of COVID, I still have an Oxford affiliation at least until April. I'd like to thank Susan and her group for the work, Lindsay for all the help with imaging, Sabine and the guys from MPI Stuttgart, so that's Klaus Kern's group, who are very active in this space, and I encourage you to check them out. They're doing some fantastic work. The guys from Thermo and our funding, especially the Queen's College, and happy to take any questions. Thank you.